Okay, everyone. What we're going to do now is we're going to enter the second third of the course. And so, if you recall, the first third of the course is basically molecular genetics. And we learned about all the mechanisms that occur in the cell with regard to all the genes, their control, protein production, and so forth. And so now what we're going to do is enter another phase of the course where this is applied genetics. What we're going to do is we're going to see a lot of the fun stuff that we do with what we've learned from genetics, where we've developed tools and applied those tools knowing how our genetics work to produce all sorts of things, whether it be medicines, agricultural products, and so forth. And so our first lecture into this journey of applied genetics is going to have to do with recombinant DNA technology, okay, or what a lot of people call biotechnology. And what we see on the screen is really the first example of biotechnology. This is an Egyptian hieroglyph, and what we can see is we can see them picking grapes on the top right-hand side and using that in the production of wine. The production of wine from grapes is an excellent example of biotechnology because it's where we use natural resources to produce a product of some sort. And so, thinking of that, now we can see how much more we can apply as the tools have become more technological and sophisticated. And these are some of the things that we're going to see in this lecture. So there are really four major revolutions in genetics, and there might be a fifth coming, we don't know. So the rediscovery of Mendel. This has to do with inheritance. We're going to see that in the third third of the course. DNA structure and the birth of molecular biology, we saw that back during our first lecture when we were looking for the structure of DNA. The third is restriction enzymes and recombinant DNA technology. That's what we're going to do today. And then the fourth is genomics. Genomics really is when computers came into the fray, when the human genome was sequenced, and we developed all of these computer tools to help us analyze, learn, and then later on create things with regard to genomes. Now, we don't know what the future holds with regard to AI's influence. And so, who knows? There might be a fifth revolution in the future where AI really does help us plan gene editing, helps us discover things of the genetic nature that we didn't know before. So, we'll stay tuned and see how that plays out. And so today we're going to do restriction enzymes and recombinant DNA technology. And this is really where we are able to take DNA and recombine it in different ways. We can isolate fragments from DNA. We can use genes that we've pulled apart away from the genome, put them into things like plasmids, and we'll discuss what those are, in order to produce proteins that we can then use for medicine or any other application. And that's what today's really focus is about. And so we're going to see how we can isolate genes apart from everything else. And not just genes, you can isolate any part of the genome, whether you're studying a gene, studying a promoter, studying an enhancer, studying a microRNA, studying other things that we haven't learned about, like long non-coding RNAs, short non-coding RNAs. There's all sorts of things embedded in our genome that we can study if we just isolated them. And so, why is it that we would want to pull out a gene out of the genome? Well, one of the things that we can do is we can have cells produce mass quantities of that. We're going to see an example of that. We can alter gene sequences and put them back into organisms, put them back into cells. We're going to talk about that too. We can produce any type of protein, whether in its original form or modified, and we can study how things are different from one species to another. And that gives us a little bit of insight into adaptation, into migration, and things like that. So let's look at what we can do if we clone out a gene, and we're going to see what that means and how we do it. How is it that we clone something out? So what I'm going to do for our example is we're going to look at insulin. Now, we probably all know that insulin is involved in diabetes. And so this is the first thing that we're going to look at when we do recombinant DNA technology. Okay, so let's take a look at physiology and let's see exactly how this works. Why is it that insulin is important? So on the right-hand side, what you're going to see is you're going to see an example of your system right after you've eaten. So let's say that you went to lunch and you had that burger and you had that soda and maybe some chips. We partially digest it by chewing it in our mouth, goes down the esophagus. It will be it will be digested even more in the stomach. It will then go down into the small intestine where it'll be digested even more so. But here in the small intestine is where we're going to do our absorption. In other words, all of that stuff that you ate for lunch, whether it's the proteins, whether it's the sugars, whether it's the lipids, in the small intestine, all of that stuff is going to be absorbed through the small intestine and into the circulatory system. The circulatory system is what is on the other side of the intestinal walls. So from eating it, digesting it, absorbing it in the small intestine, now it's in our circulatory system. And what we're going to concentrate on, instead of the cholesterol and the proteins, what we're going to look at right now is we're going to look at the sugar in that diet that you just brought in. And so what we can see is we have high glucose in our circulatory system. So we're looking at our arteries, arterioles, capillaries. And so the glucose, the sugar is extremely important because what you want to do is you want to take that glucose and send it off, make it available for the 50 to 70 trillion cells in our body. Because what they're going to do is they are then going to absorb the glucose and they are going to produce ATP out of it. And ATP really does drive a lot of the biochemistry in our cells. And so here we are after lunch, and we have a lot of glucose in the circulatory system. Now, when you look at a circulatory system, our bloodstream, right, you really shouldn't have high levels of sugar there for very long. It starts to cause a lot of problems. And we're not going to go into all of that physiology, but to avoid a lot of the physiological problems that comes from high glucose content in the bloodstream, what we have to do now is tell all the 50 to 70 trillion cells in your body, hey, start taking up this sugar. So that all the cells in your body, which make up your tissues and your organs, they start to take in all of that sugar from the bloodstream and take them into the cells, where in the interior, the mitochondria are going to use that to create ATP. But how do we do that? How do we tell all of the cells of the body, hey, can you start to take up all this sugar? Well, when you have high glucose content, that is detected by the pancreas. The pancreas has multiple functions. It has enzymes that helped us digest the food when it was in the small intestine. But here it's going to perform another task. In your pancreas, you have cells called beta isolate cells, or we're just going to call them beta cells. When high glucose is detected in the bloodstream, those beta cells release insulin into the bloodstream. And what that insulin is going to do is it's going to tell the cells of your body, take up this glucose. It's going to do that in the fat storage as well, in the adipose tissue. They are also going to take up a lot of the excess. And so, what happens with all of the cells of your body take up all of this glucose? Well, what happens is the glucose levels in the bloodstream drop back to normal levels. And that's what we want. Let's take a look at what happens in the opposite direction. That is when you have very low levels of glucose. And that happens under starvation. So what do we mean by starvation? Starvation is basically when we are fasting, right? When we fast, what that means is we are no longer eating. And when is it when we fast the longest? Well, for most of us, that would be overnight. You might have dinner around 6, 7, or 8 p.m. You might not eat breakfast till 7 or 8 the next morning. And so you're looking at around 12 hours in which you have been fasting, not eating food. So what you can see is the blood sugar levels drop in the circulatory system because the metabolism in your cells is still going. So they are still taking up sugar as they are creating ATP to drive their biochemistry. Well, we have to make sure that the blood sugar levels don't drop too much, right? Because the cells continue to need that. Your brain cells continue to need that. Your muscle cells, like your heart, which are beating, all of these cells throughout the body still need this. So the pancreas again detects this low level of glucose. And now we have a different set of cells in the pancreas that fall into action. Those are the alpha cells. And what the alpha cells are going to do is they are going to release glucagon, and glucagon signals the liver to release glucose into the bloodstream. The liver is one of our filters, but it's also a storage device. The liver is going to store cholesterol for us. The liver is going to store glucose for us. So, when we are fasting and the cells are going to need all of this energy anyway, the liver starts to release glucose, and now the glucose levels are normalized. So, what is it that happens in diabetics? Well, it depends on the type of diabetes. We're going to talk about type 1 diabetes. Type 1 diabetics suffer from an autoimmune disease. What this means is their immune system attacks part of their own body. And in this case, the immune system, for reasons we don't fully understand, attacked the beta cells in the pancreas. We know how they attack, but we don't know why. All of those white blood cells that attacked the beta cells and destroyed them, those should not have existed. We should have cleared those out before they had a chance to attack. We do that all the time, but somehow they didn't get cleared out and they attacked, and now this person has no beta cells. So they can take in sugar by their diet, and the sugar will remain in the circulatory system, and that's going to cause problems. And so, what is it that type 1 diabetics have to do? Well, they have to inject insulin, right? If they have to inject insulin, that then is the trigger for all of the 50 to 70 trillion cells in their body to take up all of this sugar from the diet. And so this started to become a huge problem. And what happened was researchers started to work on solutions, one of which is to see whether the insulin from other species worked in our bodies. And sure enough, they found that insulin from other species does work very well in our bodies. The experiments were done first with canines. So these were dogs. They tried canine insulin and it worked. And so later on, they moved to pig insulin. Why would they move to pig insulin? Well, pigs are a little bit closer to us in their physiology. But not only that, but pigs are an agricultural product. So you were going to have insulin as a byproduct from the pigs that were part of the agricultural industry. And there were a lot of them. However, we knew that as time would pass, the population would grow. Today, we stand at somewhere in the proximity of 330 million people in the US. And what we knew is there wouldn't be enough insulin. We would never have enough pigs to produce the quantities of insulin required for all the diabetics in our population. And so, what happened is we had researchers move in to clone the insulin gene and produce it in laboratories so that we had large vats and we had an immense quantity of insulin for the population. That cloning and that production is what we're going to look at. So these slides basically talk about what I just said in terms of insulin and its relationship to diabetes. The cloning of insulin and its production, it's one of the things, not the only thing, but it's one of the things that put the City of Hope on the map. And so the City of Hope is located approximately 17 miles from Cal Poly Pomona in the city of Duarte. And they are the ones that did this work. So they cloned the human insulin gene and they put it in a plasmid. We will see what that is. They introduced this into bacteria, and then the bacteria synthesized the human insulin protein. Then you could purify the protein and get rid of all the bacterial proteins. And that purified insulin is what you could package and then offer to diabetics. So it's important to note that this insulin, which is being produced by bacteria, is identical to the human insulin. So even though the bacteria are the ones that are making it, the instructions that they're using are the instructions from the human genome. So if we put a human gene into bacteria, when they make the protein from that human gene, they will be making human protein. Bacteria don't make bacterial insulin. There's no such thing as bacterial insulin. The bacteria make human insulin because the instructions came from the human genome. And so, because it's human, you have less risk of rejection by the immune system, which some people may do. They may reject the insulin. Now we can make it in unlimited quantities because we're doing it in the laboratory, and we can have massive vats of bacterial chambers, and you can purify it so it's very pure. When we do this with these recombinant proteins, they're called recombinant. We're going to see why. When we do that, it does have a research and development stage, which can be very pricey, over time that should go down. And there's reasons why insulin didn't come down for many, many years. That has more to do with business than it does with genetics. Okay, what we're going to do is look at an overview of how we clone a gene and produce protein from it. And then we're going to look at some of the details afterwards. So what we have on the screen is a fragment of DNA. It's showing us purple. This can be a gene, this can be a promoter, this can be an enhancer, this can be any part of the genome, whether human or from another species, that you are looking to either study or produce something from. So people clone areas of the genome all the time to study them. You can study genes from humans, plants, other animals, insects, bacteria, and so forth. So we have the fragment that we're looking to clone, and now we have a plasmid. Okay, so what's a plasmid? A plasmid is basically almost like a small bacterial chromosome, in that it's small, it's double-stranded, and it's circular. So this thing is much smaller than the bacterial chromosome. And so you can put genes on this thing. You can put genes, you can put promoters that control the expression of the genes. You can have multiple genes on it, and you can put that in a cell. And then those genes can be expressed in that cell, and now you have a modified cell. Okay, let's see what we're doing here. We're taking our plasmid, which again is like a little chromosome, and into it we're going to insert our DNA fragment. That's what we see here in the second step. So what we've done is we've recombined this DNA so that now we have the plasmid and we have the fragment. And now it's one piece of DNA, circular double-stranded piece of DNA. That's why we call it recombinant DNA, because we're going to start to put DNA together to function in ways that we need. And we're going to see that throughout this process. So here we have our recombinant DNA, where we have a plasmid with our piece of DNA inserted. And now what we're going to do is we're going to take that and we're going to put it into bacterial cells. Why do we put it into bacterial cells? Well, because bacterial cells can make a lot of this. So we're going to take that and we're going to insert it. In this process, there's a few things to consider. You can have a test tube with millions of bacteria, okay? And you're doing this in the laboratory. And then in another tube, you do this experiment where you insert this fragment into plasmids. And so you might have hundreds or thousands of plasmids that are teeny, teeny, tiny. You can't see them, but you have them in a tube. And you can do an experiment where you insert this fragment. We're going to see some more detail on that later. And then you take those hundreds or thousands of plasmids and you introduce it into a small test tube with millions of bacteria. And what the bacteria will do is they will start to take up the plasmids. Now, notice you might have thousands of plasmids and millions of bacteria. What does this mean? It means that the majority of your bacteria will not have a plasmid. There aren't enough plasmids to share. And that's what you see here on this step towards the bottom. You have an E. coli with its blue chromosome, and then inside you have a plasmid. It took up the plasmid. But millions of bacteria won't do that. There's just not enough plasmids to go around. And here's the thing, those bacteria are useless to me. If you are a bacterial cell, but you don't have my plasmid, you are useless to me. You're just some bacteria. And so what we need to do in the lab is we need to kill all of the bacteria that don't have our plasmids. We only want the bacteria that have our plasmids. They're the only ones that are going to help us. We're going to see why. So then what do I do with all these millions of bacteria who after some hours don't have plasmids and are useless to me? Well, it's like this. When you go to the doctor because you have an earache, the doctor says, it looks like you have a bacterial infection. What does the doctor prescribe you? The doctor is going to prescribe you antibiotics. Antibiotics kill bacteria. So this is what we're going to do. In that test tube, in that test tube, what I'm going to do is I'm going to introduce antibiotic. But wait a minute. We just said that the antibiotic is going to kill all the bacteria. Well, that doesn't sound good because that's the end of your experiment. Let's go back. Let's look at our plasmid. Notice there's something on the plasmid. It's depicted here in yellow. This is an ampicillin resistance gene. So whichever cell has this gene, it will be resistant to ampicillin, which is an antibiotic. So what I'm learning about my plasmid is not only does it have my gene of interest, but it has an ampicillin resistance gene. So now if we introduce thousands of these plasmids to millions of bacteria, the thousands or hundreds of bacteria that take up a plasmid will be resistant to the ampicillin antibiotic that I just introduced into the bacteria. The cells that don't have the plasmid, the ones that are useless to me, are going to die from the ampicillin, from the antibiotic. So that if I take the bacteria, I introduce the plasmids, then I let them grow overnight in the test tube. I can also do that on a petri dish. I'm not going to worry about the details of how to do this, but the point is this. If you have my plasmid, you live. If you don't have my plasmid, you die. And overnight, two things are going to happen. We see that over here on the right-hand side. Inside the bacteria, the plasmids, which are like these little tiny chromosomes, are going to start to copy themselves. And now there's more. On top of that, the bacteria are going to start to divide. And now there's more bacteria. So on the screen, look how many copies of my plasmid I have. In other words, look at how many copies of this DNA fragment I have. Because I have so many copies, this is why we call this cloning. As we know, clones are copies of something, right? So we just cloned this DNA fragment. And it might be a gene, it might be a promoter, it might be an enhancer. It all depends on the project that that laboratory scientist is involved in. So we just cloned that piece of DNA. So there's different types of inserts. It can be from genomic DNA, and we're going to see that. It can be from messenger RNA, and we're also going to see that. Our vectors are two kinds. We just saw bacterial plasmids, like little bacterial chromosomes, but people can also use viruses. We all remember lambda from a previous lecture. You can take a virus like that, delete the bad genes, like the virulence genes, which allow it to spread, and put in the genes that you want, and those viruses will then introduce your genes into cells. So we can do plasmids, like we just saw, or we can use genetically modified viruses to do the same thing. A library, we're going to talk about that in just a little while. Okay, so let's use all of this and see how you would you would discover a gene, you would look for a gene. In other words, if there's a function in the body and you're trying to look for the gene that does that, how is it that you would do that? Well, there's many different ways. We're going to look at one here. So let's say you're working with mice, because in mice you can do experiments that you can't do in humans. They're mammals, right? So we can do mouse experiments where the mice have some sort of human-like disease because they've been genetically engineered that way. So we use the mouse models for a lot of things. But what I first have to do is I have to figure out where the mouse gene is because I'm going to study that. You can also do this for humans, and so you would use human cells to do what we're about to do. Let's take a look. So here we have Mr. Mouse sitting here, and what we're going to do is we're going to extract DNA from the mouse. And usually what they do is they clip the tail. You clip the tip of the tail. There's no nerve endings there. When you clip the tip of the tail, then what you do is you do exactly what you guys did with strawberries. Now, there's some modification to the protocol, but it's not that different. What is it that we did? We took the strawberries, we we basically dissolved all of the bacterial cells, and the DNA came out, and you were able to isolate that with some salt, with some ethanol. Again, this protocol would be a little bit different, but extremely similar. So you take your mouse or your human cells, if you're using human cells. In this case, you're going to clip the tail. You're going to extract the DNA from it, just the way you guys did. Okay, what's going on over here on the other side? Now we're going to get some plasmids, and we just buy these from biotech companies. Biotech companies sell us bacteria that inside are carrying plasmids. So this is how they sell them. So when they get to the lab, what you do is you just do exactly what you did with strawberries. You take the bacteria, you let them grow once you have a lot, and that just takes overnight. Then you pop them open and isolate all of the plasmids. That's what you guys did. You popped open the strawberry cells and isolated the DNA. So we're doing that here. So now we've got the genome from either human cells or this mouse, and you've got plasmids. And what we're going to do is we're going to put them together, and I'll explain why. Okay, this is a genome, but this thing is massive. It is massive. It's got millions of base pairs. It's too big to clone in something so small like a plasmid. Remember, a plasmid is like a tiny bacterial chromosome. As a matter of fact, the bacterial chromosome is many times bigger than a plasmid. So how are you going to clone this whole mouse genome into a tiny plasmid? It doesn't work. So what we're going to do is we're going to take that DNA and we're going to fragment it. We're going to fragment it. And then we're going to take those plasmids and we're going to open them up. We're going to cut them open. And what we're going to do is we're going to insert all of these fragments into the plasmids. So this graphic here on the bottom, it is the same as this graphic here on the top. Okay, so we took the genome from either the human cells or the mouse. We isolated the genome and we cut it apart. And now we opened up the plasmids. These are in two different test tubes, and we take some of the fragments of the genome, we take some of the plasmids, and we put them together. And we've inserted different pieces of the genome into different plasmids, so that when you take all of the plasmids together, you have the entire genome. But each plasmid only has one piece. So the collection of plasmids, that's what we call a library. A collection of plasmids that when you put them all together, they contain the entire genome. But each plasmid only has a small piece of the genome. What we then do is we take these plasmids and we introduce them into bacteria and we let the bacteria grow overnight, right? And what we're going to do is we're going to use some assays to figure out which bacteria is carrying the gene that I am trying to discover. What do those assays look like? Well, there's hundreds of them. It just depends on the function of your gene. There's other ways that you can look for it. We're not going to go into that, but you will learn about much of that if you take some of the 4,000 level classes in molecular biology. Okay, so we cut up the genome and we stuck it into all of these plasmids. And you might be thinking, okay, but why again? Is it because the genome was so big? Yes, that's part of it. But I want to give you another example. Let's say I have a book and it has 3,000 pages on it, right? This book has 3,000 pages in it. And I am looking for one specific page. Not only that, I'm looking for one specific sentence. I've got a 3,000 page book, and I'm looking for one particular sentence. Let's pretend that that sentence is select or screen clone with gene of interest. I only chose that sentence because it's on the screen. Now, I don't have a digital copy of this book. I don't have a PDF. I only have a physical book, which is made out of paper. And I give you this book, and it's got 3,000 pages. And I said, you know what? I need you to find this particular sentence in the book. The sentence is select or screen clone with gene of interest. I'm going to go to lunch and I'll be back. And you're sitting with this 3,000 page physical paper book, and now you've got to go through it and find this sentence. It's not going to be in the index, it's not going to be in the table of contents. You've got to go through it page by page. And if you miss it, if you get to the end and didn't find it, you're going to have to start all over again. How long is that going to take you? It's going to take you a long time, right? So you know what? I just don't think I like that approach. Here, let's do something different. I know. I'm going to bring in 3,000 students. I'm going to bring in 3,000 students. And to each student, I'm going to give one page. I've got 3,000 students, and they're each holding one page. And I tell all the students, I need you all to find a particular sentence for me. The sentence is select or screen clone with gene of interest. And each student has one page. How long is it going to take us to find that sentence? Chances are it won't even take 5 minutes. Chances are depending on where it is on the page, chances is going to take you 1 to 3 minutes, maybe, before you find that sentence. So look at that. I divided up the book, I gave it to students in small sections, and we were able to find our page very quickly, or our sentence. We were able to find our sentence very quickly. That's the reason why we're doing this. I don't know where that gene is in the mouse genome or the human genome. And so what I'm going to do is I'm going to cut it up and I'm going to stick fragments in these vectors. These are my plasmids. And then I'm going to put those in bacteria. So the bacteria serve the role as students in the examples. And the DNA fragments that were inserted into the plasmids, somewhere there in one of those plasmids is going to be my gene, but I don't know where it is. But what I do know is that one bacteria has it. One bacteria has it. And all I've got to do is screen the bacteria for that particular function. Or there's other ways to screen it, as I've talked about, which we won't go into this class. And that's how we discover a lot of genes. We clone them, and then we assay for them. Let's go into some details. So I said I cut up the genome, and I said I opened up the plasmids, but I didn't really talk about how. So we're going to talk about something that revolutionized biology. It brought us into the era of molecular biology, which we're still into today because we do molecular biology everywhere. See, what we discovered is that many prokaryotes have what we call restriction enzymes. So many bacteria, in other words, many bacteria have enzymes that cut DNA. The screen says over 400, but we are in the thousands now. We have discovered so many enzymes in bacteria that cut DNA. But these are called restriction enzymes. Why are they called restriction enzymes? They're called restriction enzymes because even though these enzymes can cut DNA, they cannot do it randomly. They have specific target sequences that they cut. For example, here we have an enzyme on the screen called EcoR1. Comes from E. coli. EcoR1 will cut DNA, but it won't cut it just anywhere. It is restricted. It is restricted as to where it can cut. So where can it cut? EcoR1 will cut the DNA if it ever sees its target sequence, or what we also call its recognition sequence. When it recognizes its sequence, when it sees its target, it will cut. And for EcoR1, the target sequence is GAATTC. That is its target. And for most of restriction enzymes, their targets are palindromes. Well, what is a palindrome? A palindrome is something that reads the same forwards as backwards, like race car, right? And so what we see here is that the recognition sequence by EcoR1 is GAATTC. What is the sequence on the other DNA strand? Well, if we follow our rule of C's bind to G's and T's bind to A's, then you'll notice that the recognition sequence on the other side is GAATTC. What this means is it doesn't matter which side of the DNA the enzyme is reading. It's going to find GAATTC right there on that spot. And it doesn't matter if it was reading the top strand or the bottom strand. It's going to cut. And that's what we see here. And we see that it cuts. Now, some enzymes cut right in the middle, but most enzymes don't. Here, EcoR1 cuts between the G and the A. So we see that on the top strand, it cut between G and A, and we see on the bottom strand, it cut between G and A. And so we have overhangs, right? We have 5' overhangs. We also call these sticky ends, and we're going to see why we call them sticky ends later. Okay, let's take a look. So here we have our plasmid. And what we're going to do is we're going to take that plasmid, which we bought from a company. We're going to put it in a test tube. In that test tube, we're going to add some EcoR1 enzyme, which we bought from another company. And it's going to cut between the G's and the A's. It's going to linearize this plasmid. It's going to open it up. And that's what you see there. And you can tell they have these overhangs. We're going to call those sticky ends. We will see why. How often does equal or 1 cut? Well, if I ask a second question, how big are the fragments that equal or 1 creates? Those two questions can be answered by the same math. Okay, let's look down here. So equal or 1 has a recognition site. How many nucleotides, how many base pairs long is the recognition site? Well, GAATTC. There's six. Six nucleotides. So, how often will equal or 1 find its target site if we let it cut the human genome or the mouse genome or the strawberry genome? Let's take a look. What is the probability that the first nucleotide that equal or 1 finds is a G? Remember, we're looking for its recognition sequence. Well, the chances that it find a G is 1 in 4. Okay, what are the chances that the second nucleotide right after that is an A? Well, it's also 1 in 4. We only have 4 nucleotides. And what about the next one, and the next one, and the next one? So at all 6 positions, the probability of having the correct nucleotide is 1 in 4. And so if you multiply that out, you get 1 over 4096. What this means is equal or 1 will find its recognition sequence. It will find its recognition sequence every 4096 nucleotides or so on the genome. And that's where it will cut. So the fragments that it's creating are on average 4096. Now remember, these are averages. In other words, equal or 1 might find its sequence 30 nucleotides apart, not 4,000. So the fragment that you create is 30 base pairs. It might be walking on some chromosome, and its recognition sequences might be 28,000 base pairs apart. So the fragment that it creates is 28,000 base pairs. So you see, it's only an average. So when you do this, you actually get a range of fragments. But if you average them out, it's about 4096. Okay, so test tube 1 has my plasmid. I bought that from the biotech company. I take some of that, put it in a test tube, throw in equal or 1, and it cuts it open. And it is, remember, cutting between the G and the A. So it's giving me some sticky ends. Those sticky ends have a sequence of TTAA. Now I'm going to take either the human DNA from our cells or the mouse DNA genome from the mouse. And what I'm going to do is I am going to take some of that in another tube and I'm going to cut that with equal or 1. It's going to cut that genome wherever it finds its recognition sequence. And we're going to get different sizes fragments. But notice they also have sticky ends because that's the way that equal or 1 cuts. And now what I'm going to do is I'm going to take another test tube. I let the equal or 1 cut for 1 or 2 hours. Then what I'm going to do is I'm going to take a little bit from test tube 1, those are my plasmids, a little bit from test tube 2, those are my genomic fragments, and then I'm going to put them in a third test tube and let them come together. And what happens? Well, because they were cut by the same restriction enzyme, their sticky ends are the same. And so what am I going to have? I'm going to have those fragments stick onto the plasmids. And then I can take a little bit of ligase, I can throw that in the tube, and ligase is going to glue that together. And what I have is I have a plasmid with my insert. So we're cloning all of these fragments. Now, you might think, well, could the plasmid close in on itself and just have no insert? I mean, you cut it open, can it just close on its own? It can. Some plasmids can close on their own, but a lot of them are going to have these inserts. Remember, all of them together form a library. That is how, that is how we can produce a library from genomic DNA. Now, if we're looking for a gene, we can look for it on the DNA, but the DNA is massive. It's an ocean of DNA. In humans, 6 billion base pairs, right? Or 3 billion base pairs, but 6 billion from each cell that you're popping open and taking DNA out of. And so some research or some researchers, they decided we don't want to work with DNA because only 1.5% of it actually has coding regions for protein. So what we're going to do is we're going to work with the messenger RNA that is floating around inside the cells. Because if a cell is expressing the messenger RNA, it must be important. You're not going to take DNA and transcribe it to give you messenger RNA if it's not important to you. So what they did is they said, we are going to isolate the messenger RNA instead of the DNA. And we're going to clone all of that. Now, messenger RNA obviously is RNA. So what we're going to do is we're going to have to convert that to DNA. In other words, we're going to take the messenger RNA and we're going to make the complementary DNA of the RNA. We're going to make the complementary DNA of this RNA. And so that's why we call it cDNA. That's the complementary DNA of RNA that we isolated. Because plasmids are made of DNA. We can't put RNA inside a plasmid. So we have to convert it. We have to convert it to DNA. So what they did was with a slightly modified protocol, instead of isolating the DNA, you can isolate the RNA. And we've got to convert it to DNA. So how do you do that? It's actually very simple. We know that messenger RNA has a poly A tail. Remember that? There's a bunch of A's here at the end. So what they did was they made a bunch of poly T's. These are your primers. Everyone remember DNA polymerase? It looked for a primer, and then it grabbed it, and then it made DNA. So in this case, we isolate all the messenger RNA from the cells. We throw in poly T's, hydrogen bond to the poly A tails, and then we use a special enzyme that is going to take messenger RNA and make DNA from it. We can't do that. As humans, we go the other way around. We take DNA and we make RNA. We cannot take RNA and make DNA, but other organisms can. As a matter of fact, viruses can. So from viruses, we borrow this enzyme. It's called reverse transcriptase. So it's going in the other direction. So in this test tube, I'm going to throw in all of the messenger RNA that I got from those cells. I'm going to throw in lots of poly T primers. They're going to bind to the tails. I'm going to throw in reverse transcriptase, and reverse transcriptase is going to create a DNA strand from that messenger RNA. That is my cDNA, my complementary DNA. And something curious that reverse transcriptase does is it actually adds a couple of nucleotides at the end. We don't know why it does this, but it does. And these nucleotides match the nucleotides that it created from the end of the messenger RNA. Why is that important? This is why. In this test tube now, I can destroy the messenger RNA. That's very easy. And these nucleotides roll over and create a primer for me. So now I can throw in a DNA polymerase. It's going to see that little primer and it's going to make my double strand. It's going to create my double strand DNA. I'm going to use a nuclease, which cuts nucleic acid. This specific nuclease cuts off that end. And now I've got double-stranded DNA that came from RNA. So this is my double-stranded cDNA. And what I can do is the same thing. I can take it, I can treat it with EcoR1. EcoR1 will create fragments. I could take those with their sticky ends and I can insert them into plasmids. So whether I'm using genomic DNA, which was the entire genome, or I'm using cDNA, which is the collection that came from all the messenger RNAs swimming around in these cells, I can clone all of this. Here's the overview. Here's the DNA. Regardless of where it came from, it can be genomic or cDNA. I can take that. I can fragment all those pieces of DNA. I can take my vectors, the plasmids that I bought from the biotech company. I open them up and I insert all of these fragments. Now I've got my library. Each plasmid has a different fragment. I take all of those, I introduce them into bacteria. One of these bacteria is going to have my gene. So notice what we have here. This bacteria didn't take up a plasmid. There weren't enough. That bacteria is going to die because of the antibiotics. This bacteria has a closed plasmid. It closed up on itself, which it can do. This plasmid has an insert. This plasmid has an insert. This one closed up on itself. There's going to be a lot more plasmids which have inserts. I have these bacteria in a test tube. I throw in my library. After a while, maybe about an hour or so, I take all the bacteria, I put it on a petri dish, I let that grow overnight, and the next morning I have these colonies. Each one of these colonies originated from one bacteria that landed there. So one bacteria lands here, and if you stick this petri dish overnight in an incubator, the next morning you'll have about 10 million bacterial cells. They grow so fast. And then that's what you see. You see this little colony. Each one of these colonies is about 10 million bacterial cells that came from that one original one. So this is more like a Xerox machine, right? This is a copier. These 10 million all have the same insert. These 10 million over here, same insert. These 10 million over here from that colony have the same insert. So it's like a Xerox machine. Now I'm just copying what I have. But each colony has the same insert. Remember the pages. Remember the pages that I gave you as students. We're looking for this sentence. We're looking for this gene. And now I can take those bacteria from that colony and I can put it in a flask. This is a flask with media. This is just a liquid with a lot of yummy nutrients for bacteria to grow in. And I grow them here. And then the next day I have hundreds of billions of bacteria. All the billions of bacteria in this flask have a single page. They're all the same. But this flask over here has hundreds of millions of bacteria with a different page. And so I have cloned many areas of the genome. Actually, I cloned the whole genome, but not in one plasmid. It's too big. So this library is a collection of clones, where together they have the entire genome and now many copies of the genome. As I said, there's a way to assay which one of these has my gene. Again, there's hundreds of assays. It just depends on the function of your gene. And then what you do is you say, okay, I need to isolate the plasmid from the flask that was positive in my assay. So I tested them, and let's say this top flask here is the flask with all of the bacteria that have my gene in it. So now what I want to do is I want to purify out that plasmid, take that DNA and send it to a sequencing facility. That sequencing facility will then sequence it and send me back the sequence of my gene. So this is what we're going to do. Here's our flask, and here's all of our bacteria, and we did whatever biochemical or other type of assay that was needed, and we know, this is the flask with the bacteria that has my gene in it. And we're going to do exactly what you did with strawberries. We're going to take those, we're going to spin them down, and then we're going to lyse them, which means pop them open, and all the guts come spilling out. And then we're going to isolate our plasmids. Our plasmids are DNA. They're smaller than the genomic DNA. So there's there's a way to actually separate these. And now I need to clean all those plasmids. I need to clean them so they don't have all the bacterial gunk from all the dead bacteria. So this is what I am going to do. What charge does DNA have? That's right, DNA is negative. And so what I'm going to do is I'm going to take all of the floating part of this solution because I centrifuged the tube, so all the genomic DNA, the proteins, everything comes down here. I take everything that's floating, that's where my plasmids are going to be, and I load it into a little column like this. Now, this column has a bunch of beads in it, and those beads are positively charged. So that if I take all of this that's floating and I put it in the column, everything is going to run through. But the positive beads are going to grab onto the negative plasmids. All of the other junk is going to come through, and then I can wash it. I can take some nice buffers and or even water and just wash it so that all I have now is nothing but beads that have grabbed onto my plasmids because their charges are the opposite. All the junk has come through and it's gone. And now what I do is I take some salty water and I add the salty water to the column and all my plasmids come out. Well, why? Plasmids are DNA, negative. The beads are positive charged. So those two stick together. But if I add salty water, when the salt comes through, what is salt? Salt is sodium chloride, a bunch of positive ions and a bunch of negative ions. So what happens is the sodium, which is positive, grabs on to the DNA. The chloride ions, which are negative, grab on to all the beads. And so you have more salt now grabbing onto the DNA and the bead, and the DNA comes off and it just slides out. We can kind of see a zoom of this. Here's our column. Here's our DNA over here. The phosphates are what make it negative. Here's our bead. It's positively charged. Over here, we see the DNA. And we see the beads. And they're loving being attached to one another. Here comes the salt. And the salt is going to grab onto both the DNA and the beads. The DNA now lets go of the beads and it flows out. And so that's how we purify the plasmids, and now we can take that, send it to a sequencing facility, and they'll sequence it and tell us what it is that we have. And you'll know your A's, C's, G's, T's. You'll know the sequence. So what we're going to do now is we're going to do some restriction mapping. We're going to do some restriction mapping. When we clone things, we need to know what enzymes can cut those plasmids and where they can cut those plasmids. So we're going to do some restriction mapping. Here's how this works. So you work for a biotech company and you are producing plasmids. And so you need to give your customers a map of your plasmid to show them which enzymes cut it and where the enzymes cut it, so that I can look at the map and go, hey, that's a plasmid that I can use. So this is a particular plasmid called PGEX. P stands for plasmid. And so this is PGEX. It's a plasmid that's sold by a company. And so what they had to do is this. We take PGEX and we're looking to see where different enzymes cut. So we're going to concentrate on two enzymes, PVU1 and BAMH1. We take some of the plasmid and we put it into three different tubes. PVU1 is allowed to cut for an hour at 37 degrees Celsius. Why 37 degrees Celsius? Well, if you take Celsius and you convert it to Fahrenheit, you'll notice that that's 98.6. And so this is body temperature. That's where these enzymes function the best. So 37 degrees Celsius for an hour. Now what you're going to do is you're going to take some of that plasmid, you're going to put in another tube, and you're going to put in a different enzyme. This is your BAMH1. And then in the third tube, you're going to put in some DNA, and then you are going to put them both in. You're going to let those enzymes cut for an hour, and then you're going to run this on a gel to see the sizes. If we don't remember, gel electrophoresis is a technique where we can load DNA onto a gel, and then we turn on an electric current. And because DNA is negative, it's going to run towards the positive end. It's going to run towards the positive end. And the smaller the piece of DNA, the faster it's going to go through that matrix. The larger the DNA, the slower it's going to go. So we can actually see the fragments of the DNAs. Okay? Let's take a look at what happened to our reaction. Here's my enzyme. I'm sorry. Here is my plasmid. I throw in PVU1 and BAMH1, and there's a bunch of other enzymes that I'm going to have to do later on. But this is what happened. Now, PGEX is about 5,000 base pairs. I take that reaction. Remember, I put this in a tube and I threw in PVU1. That's an enzymatic reaction where it's chopping up the DNA. I take that reaction and I run it on the gel. And what do I see? I see a band that's 2,800 base pairs, and I see a second band, which is 2,100 base pairs. Now I take my other test tube where I had BAMH1 and I run that on the gel. And I have a band, I only have one, and it's 5,000 base pairs. And then I take the reaction that had both enzymes together, and I have three bands. I have one that's 2.8 kilobases or 2,800 bases. I have one that's 1,200 bases, and I have one that's 800 bases, right? Base pairs. What is this telling me? It's telling me that PVU1 cuts this plasmid and produces a 2.8 kilobase fragment. That BAMH1 cut it only one time, which is why it just has one fragment, which is the entire plasmid. But if I throw in both of them together, what it tells me is that what BAMH1 did is it cut once again, but where it cut was somewhere in this 2100 base pair fragment. How do I know that? Because the 2100 base pair fragment is gone. Notice how it's not there anymore? Where did it go? BAMH1 cut it. It cut it and gave me a fragment of 1.2 kilobases and 800 bases. And so I start to build a map like that. And I'm going to do all of these other enzymes as well. What we're going to do now is we're going to map a piece of DNA. And it's like doing a puzzle. Okay, what do we have here? We have a piece of DNA. And what I'm trying to do is I'm trying to map where these three enzymes cut. I've got ECOR1, Hindi3, and BAMH1. And I have experiments where I put them in combination. Notice my fourth tube has ECOR1 and Hindi3 together in the tube. And then what we have is we have what I got from my gel. Here are the size of the fragments that I got. And now we've got to put this map together. Now, what we saw previously was a circular piece of DNA because it's a plasmid. Here, instead, what I want to do is I want to do a linear piece of DNA. So you see what that looks like. Okay, let's start. I have a piece of DNA. I throw it in a tube and I throw in ECOR1. And what it does is it gives me two fragments. ECOR1 cuts and it gives me a fragment of 200. And then it gives me a fragment of 700. Okay. What I do is I take that piece of DNA and I throw it in a different tube. And in it, I throw Hindi3. Hindi3 cuts that piece of DNA. And what it gives me is it gives me two fragments. One is 600. The 600 means base pairs, DNA base pairs. Okay. And the other fragment is 300. And then for my third reaction, I take this piece of DNA, I throw in BAMH1. BAMH1 is going to cut twice. That's why I'm getting three fragments. I'm getting one that's 50 base pair, one that's 350 base pair, and one that's 500 base pair. And I'm not going to care if you can draw this to scale, but the logic has to make sense. And so I want to point something out to you. If you have a circular piece of DNA and you cut it once, you have one fragment. If you have a linear piece of DNA and you cut it once, you have two fragments. So when you're doing the maps, it's not exactly the same. It's the same process, but the fragment number is going to change. Again, if you have a circular piece of DNA and you cut it twice, you're going to have two fragments. If you take a linear piece of DNA and you cut it twice, you're going to have three fragments. And so with BAMH1, we have three fragments. Okay, how do I figure out this map? Okay, a couple of things here. I need to know how many cuts. I need to know how many cuts I need to map. How many cuts does EQR1 make? One. How many cuts does Hindi3 make? One. How many cuts does BAMH1 make? Two. I add those up. I add those up, and I need to map four cuts. I'm going to need to map four. By the time this is all over, I'm going to map four cuts. Okay? Okay, where do we go from here? This is what we're going to do. We're going to do our double maps. And when we have a double map, we're going to go back to the single maps for comparison. Let's take a look. Whoops. Okay, here we have our double map. And this double map is going to be EQR1 plus Hindi3. When we put those two enzymes in the tube with the DNA, what do I get? Well, I get three fragments. I get a 100, I get a 200, and I get a 600. Remember, these are base pairs because it's DNA. Okay, I need to know which enzyme cuts these are. So I have to go back to the single maps. So here's our next step. We are always going to take our single maps and overlay it on top of our double maps. Let's do that. Here's my single map for EQR1. I have a 200 and I have a 700 base pair fragment. I'm going to look at my double map. Can I get a 200 and a 700? I can. Of course, what I'm going to have to do is move some of this around. I notice that I have a 100 over here and a 600 over here. If I put those together, I can get a 700, which is what EQR1 gives me. And I have a 200 left over, which is what EQR1 gives me. So this is what I'm going to do. I'm going to rearrange this. And what I'm going to do is I'm going to put my 200 over here. Remember, I'm not worrying about whether you can draw this to scale. So I'm going to put my 200 here. I'm going to put my 100 here. And I'm going to put my 600 here. So notice I just rearranged my double map. Okay, I go back to the question. Looking at my single map, I need a 200 and a 700. Can I get that? There it is. I've got my 200 and EQR1 would cut here, and then I get my 700. Awesome. Now I'm going to overlay my other single map. Here it is here. Hindi3. Hindi3 gives me 600 and a 300. Can I get that? I look over here. Here's my 600. There's my 300. So my Hindi3 cuts here. My first double map was wrong. I fixed it. There is my double map. Now, you see, it's all just puzzle solving. Now what I'm going to do is I'm going to do my next double map. Here's my DNA. And what did I do with my next one? I took that DNA, I threw it into a tube and I put in Ecor 1, and this time I put in BAM H1. What do I get? Well, it looks like I get 4 fragments. So I get a 50. Remember, that's 50 base pairs. I get a 150. I get a 200. And I get a 500. Okay. Is that the right order? And are those my cuts? Let me take a look. Once again, I'm going to overlay the single maps. I'm going to overlay the single maps. So what's my first single map? Equor 1. Here it is over here. I get a 200 and a 700. Can I get that? Well, it looks like I can. My 50 and my 150, that gives me a 200. My 200, my 500, that gives me my 700. So that part may be right. So I'm going to put my E here. My Ecor 1 cut, I'm going to put there. That gives me my 200 and my 700. What do we do? Same thing. Now I'm going to take my BAM H1 single map and overlay it onto my double map. What do I get? I get a 50, a 350, and a 500. Can I get that? Here's my 50. Look at that. My 150 and my 200 give me a 350. And there's my 500. So it looks like this one, this double map, we got right from the start. So we didn't have to move anything around. Okay, now I'm going to do my third and final double map. So what's this one? This one is Hindi 3 and BAM H1. Those are the two enzymes that I threw in. Okay, what do I get? I get a 50, a 100, I get a 250, and I get a 500. So what are these cuts, and is everything in order? Same strategy, overlay the single maps. Okay, here we go. With a Hindi 3, I get a 600 and a 300. Can I get a 600 and a 300? I can, but not in this order, not in the order they have these in. So if I swapped 100 and 250, I can get a 300 fragment, and I can get a 600 fragment. So let's see if that works out. I'm going to leave my 50 where it is. I'm going to swap. So here's my 250 base pair fragment. Here's my 100 base pair fragment. And here's my 500. Okay, let's overlay again. I go to my BAM H1. I've got a 50. There's my 50. BAM H1 cut there. Actually, we had done the Hindi 3 first, didn't we? Okay. Let's keep going with BAM H1, actually. Actually, Nix that. We're going to finish Hindi 3. Hindi 3 gave me a 600 and it gave me a 300. Do I have that now? Here's my 300 here. Here's my 600 here. So Hindi 3 can cut here, and it will produce a 300 and a 600. Now let's do BAM H1. I do BAM H1, and I need a 50, a 350, and a 500. Let's see. Here's my 50. Here's my 350. Here's my 500, which means my cuts are correct after I overlay my single maps. So now I have all of my maps, 3 single maps, 3 double maps. And what I have to do is map 4 cuts. 4 cuts. All right. Here we go. I have to choose where I'm going to start. And so what I'm going to do is choose one of my double maps. I'm going to start with a double map, and I'm going to try to finish from that. So the question is, which double map should I start with? Well, I know I have 4 cuts. Let me see. This double map has 2 cuts, so I need to figure out 2 more. This top double map over here has 3 cuts. So this one's even better because I only have to figure out 1 more cut. This double map here also has 3 cuts. So I only have to figure out 1 more cut. So you can start with whichever one you want. And I usually ask the class, which one do you want to start with, the top one or the bottom one? And I let you choose. So let's just go ahead and start with the top one, but it wouldn't matter if you started with this bottom one here. That would be just fine. So I'm going to take this top one, and what I'm going to do is I'm going to draw it right back down here, exactly as I see it. I've got a 50, a 150, I've got a 200, and I've got a 500. Now I put in the enzymes that cut it. Okay, here's a map with 3 cuts. I'm missing one. So what am I going to do now? Now that I have my start point, I'm going to overlay the other 2 double maps. So who am I missing? Let me see. I have BAM H1, I have equal 1. It looks like I'm missing Hindi 3. So I need to overlay this double map, which has Hindi 3, and or this double map, which has Hindi 3. Let's take a look. And you can look at either one of them first. Let's do this one over here. On this one, I have a 200 fragment, and then I have an equal or 1 cut. Okay, let me see. I should have that here. Here's a 200 fragment, right? 50 plus 150, and here's my equal or 1. Cool, looks good. 100 base pairs after equal or 1, I have a Hindi 3 cut. Here's my equal or 1, so I'm going to walk 100 base pairs, and I'm going to put a Hindi 3 cut right there. That Hindi 3 cut then leaves a 600 base pair fragment. Here's my Hindi 3 cut. This fragment here turns out to be 100. I know that because I got that from here. Equal or 1, you walk 100, then cut. Equal or 1, walk 100, then cut. Now, it just so happens that I cut this 200 base per fragment. But if I was walking inside this fragment and I cut at 100, then I know that this fragment here is also 100 because these two have to add up to 200. So my Hindi 3 cut here is 600 on this map. If I cut it here, I've got a 100 and a 500. There's my 600. Looks pretty good. Let's do the other map just really quick. So I should have a 50 and a BAM H1, 50 BAM H1. I should have a 250 and a Hindi 3. So let's see. Here's 150 plus 100. That's 250. Sure enough, there's a Hindi 3. Then I've got a 100 and I should have a BAM H1. There's my 100 and BAM H1 and a 500. So there it is. There are my four cuts. And so what I'm going to do now is I'm just going to clean it up because it looks like a little bit of a mess. 50. And I get a BAM H1 cut. 150. And I get an EcoR1 cut. 100. And I get a Hindi 3 cut. Another 100. And I get a BAM H1 cut. And that leaves me with a 500 base pair fragment at the end. And that is my final map showing where these enzymes cut and the fragments that I would get. And so we have a lot of practice problems doing this, both with linear DNA, which is what we just did, and circular DNA, which is what we saw on the previous page, where we're trying to figure out the restriction enzyme map. Okay, what we're going to do is we're going to talk a little bit more about this recombinant DNA technology. And it has to do with one of the most amazing tools ever, which is PCR. Some of you may have done PCR. It stands for polymerase chain reaction. Maybe you did it in one of your school labs. And what it is it's an artificial DNA replication technique that allows you to take a little bit of DNA and make a lot of it. It also allows you to then amplify this DNA in the region where you have a gene. And if you take PCR and amplify a gene, then you can clone it. And so that's what we're going to look at. Let's say you want to clone a gene because you want to insert it into some cells, or maybe you just want to study it. Here we're inside a cell, and this is the nucleus here. And look at all this DNA. So notion in DNA looks like spaghetti. But the gene you want is right here in this area. So you don't want to amplify the whole genome. That's massive. You just want to amplify the DNA in that little area because that's your gene. So if we zoom in, what do we see? We see GATCs everywhere. And so what we're going to do is depict it like this. Here's the beginning of your sequence, and then you've got thousands of nucleotides. And then here's the end of your sequence. So this sequence might be, I don't know, 3,000, 4,000 maybe. And you want to amplify that region because your gene is there, that's your gene, and clone it into a plasmid. So, how do you do that? Remember how our cells replicate DNA? How do they make copies of DNA? They set down primers. So that's what we're going to do. Okay, here's that same sequence from the previous slide. Here's the beginning of the sequence. This is the 5' end. Then there's thousands of nucleotides. And here's the end. And there's the 3' end. Here's the complementary strand on the other side. And what you want to do is you want to amplify all this. Because then if you have all those copies, you can clone them into plasmids and now we can study that gene. I need primers so that my polymerases know what to copy. So what I'm going to do is I'm going to go onto the website of a biotech company and I'm going to tell them I need primers of this sequence. They'll make those and send them to me in tiny little tubes. But they're going to say, what's your primer sequence? And we'll synthesize it for you. Here we go. I'm going to take this double-stranded piece of DNA where my gene is, and I'm going to open it up. This top black strand and this bottom black strand, that's my double-stranded DNA. I've opened it up. My primers go in 5' to 3'. Remember, that's how we synthesize DNA. Here's my first primer. C, C, G, A, and so on. This is my first primer. Primers are usually somewhere between 18 and 22 nucleotides. But we're just going to say this is my primer here. Now I'm looking at the other side. I want to amplify this. Here's my other primer. G, A, T, G, and so on. If we look at this in cartoon form, it looks like this. Here's my DNA. One primer is going to latch on here on the left. That's the green one. The other primer is going to latch on here on the right. That's the blue one. And my polymerases are going to amplify everything in between these two primers. Remember, this primer tells one polymerase, hey, copy this region. And the other primer tells DNA polymerase, hey, copy this region. And so we're going to copy everything in between. This means we don't have to copy the whole genome. We put our primers exactly where we want to amplify DNA, and we're going to make lots of copies of that. And now let's do some PCR. Here's how it goes. I've got a little tube, a little test tube. I'm going to throw in my original DNA. This might be the whole genome that I got from the strawberry or from the human cells or from the mouse. I'm going to throw in my primers that the biotech company designed for me. I'm going to throw in nucleotides because I have to have nucleotides to build new DNA. And I'm going to throw in a polymerase. We're going to talk about this polymerase in a moment. Okay, when I think about DNA replication, what do I need? I need the DNA, I need the primers, I need the nucleotides, I need a polymerase, I need a helicase. Well, enzymes are expensive. So if we can go without them, let's go without them. In this case, what we're going to do in our test tube is not put in a helicase. We're not going to put in a helicase. What we're going to do is we're going to use temperature to separate the DNA strands. That's much cheaper. Let's see what happens. We're going to throw all of this in a test tube. And then I'm going to increase the temperature to about 95 degrees Celsius. Notice that is almost boiling. That is almost boiling. And what that does is the solution is so hot and the DNA strands, because of all of that energy, right, are vibrating so much that it overcomes the strength of the hydrogen bonds. It's too hot. The hydrogen bonds cannot keep the strands together. And so we denature the DNA. That means is take double-stranded DNA and make it into single strand. Here it is here. We didn't need helicase. We just increased the temperature of the test tube, and the DNA starts to fall apart. It starts to come off of itself. So now it's single strand everywhere. Now, this is almost boiling, which means my DNA polymerase is done. Remember, our temperature is 37 degrees Celsius. If I take a human DNA polymerase and I put it into this tube, an enzyme that operates at 37 degrees, and I increase it to 95, that protein's going to fall apart like spaghetti. It's just too hot. So they did something that was ingenious. They got the Nobel Prize for this individual. He got the Nobel Prize faster than anybody else for creating PCR. What polymerase can survive hot temperatures? Well, what are the hottest environments on Earth where you see organisms? Well, it's the boiling waters of geysers. We have thermophiles. We have bacteria that can survive that water. One of the bacteria that survives that temperature is called Thermus aquaticus. Thermus aquaticus has its polymerase, which functions just fine at those really high temperatures. And so Thermus aquaticus, we abbreviate as TAC. So this is TAC polymerase. It's not human polymerase. It's not E. coli polymerase. This is TAC polymerase from Thermus aquaticus. And we need it because, look, we don't have a helicase. So we make the temperature go really high and everything falls apart. And Thermus aquaticus polymerase, which we call TAC polymerase, is just fine. Okay, we raise the temperature and everything starts to denature. And now what we do is we start to lower the temperature. We lower the temperature, and what happens is the primers start to grab onto the DNA because there's so many of them. We throw primers in excess. There's tons of them. Because your question might be, well, won't the DNA strands just come together? Not if you've got tons of primers. Now the primers are going to jump on there first because there's so many of them. We bring the temperature down. The primers start to jump on there. And now what we do is we raise the temperature just a little bit to go to 72 degrees, which is what TAC polymerase loves. TAC polymerase sees the primers and it extends. It extends the DNA strand. And now what you have is you have two strands that came from the original one strand. And now we're going to do it again. We are going to increase the temperature to denature all of that, bring it down, let the primers sit, and then TAC polymerase is going to extend the DNA strand. And now we're going to have 4 double-stranded DNA strands when we started with 1. And we keep going and we keep going, so that although we start with 1 copy, we can do this 30 times. This test tube goes through 30 cycles where it gets hot, it cools down to about 55 degrees, and then it goes back up to 72, and TAC polymerase makes a copy. Then it goes back up to 95, so that everything comes apart, goes back down to 55, primers latch on, we go up to 72, and the TAC polymerase extends those, and we keep making more and more copies. If you do this 30 times, for every 1 copy that you started with, you're going to have a billion copies. Now, this is important because think about forensics. When you go to a crime scene and if you see blood, most of that blood is useless when it comes to forensic analysis because red blood cells don't have DNA. They spit out the nucleus when they were developing. And so if you see a faint smear of blood, even that faint red smear is useless. What you want is you want the white blood cells that are in that blood. And look how few there are. So even if you have a faint amount of blood, what you can see won't help you. So you need to take those cells. You're going to pop them open like you guys did the strawberries, and you're going to amplify that DNA at the crime scene so you can analyze it using forensics, which is something we're going to do later in the course. Same thing with hair. Does hair have DNA in it? No. Hair does not have DNA in it. The only way you can get DNA when you find a hair is if that hair came out of the skin and the hair bulb inside the follicle came out with it. Hair doesn't have DNA in it. It's just made of a protein called keratin. But if you pulled out the bulb and you stare at it here on the bottom of the slide, what do you see? All these little cells that were pulled out, those are the ones with DNA. But look how little you have. So you're going to take this and just like your strawberries, you're going to put it in a tube, you're going to pop them open, and then you're going to amplify all that DNA with primers and Taq polymerase. And now you have enough DNA that you can analyze. And so this has been the beginning of our journey in recombinant DNA. We're going to do a whole lot more because we're going to start to look at DNA editing. We're going to start to look at modified organisms, and it's going to be a lot of fun.